Great. Good afternoon, everyone. Welcome to the Connect Science series. Just a couple of reminders before we get started. You can check out the website should be appearing on your screen right now if you want to catch the announcements of upcoming seminars and recordings of past seminars. And please, during the course of the lecture today, put your questions in the Q&A, not the chat. That would be great. And then at the end, I can open up the Q&A for asking questions. So with that, it is my great pleasure to introduce Dr. Mark Labarge, who's coming to us from the City of Hope, where he's a professor in population sciences. And prior to that, he did his training, his postdoctoral work in Mina Bissel's lab at Lawrence Berkeley. His lab is specializing in researching changes that occur in the microenvironment, particularly in the mammary gland during aging, and trying to understand those changes that make it more susceptible to cancer initiation. He's developed pretty phenomenal and very useful primary cell culture technologies for human and mouse that are enabling them to do functional studies on aging of mammary epithelial cells, and led them to their discovery that loss of lineage specificity in mammary epithelium with age changes the way that progenitor cells respond to their microenvironment. Dr. Labarge has received numerous awards. I'll mention a couple here. He has received the Department of Defense Cancer Research Program Era of Hope Award. So with that, I don't want to take any more time, and I turn the floor over to Dr. Labarge. Thank you again for joining us today.
Thank you so much, Sandra, and thank you to the organizers. This is a very cool forum, so very honored to present our work to you. So let me get on the right slide here. Okay, so yeah, we've been interested in the interaction between aging and breast cancer susceptibility for quite some time. Out of, sort of all the commonly discussed all the risk factors for breast cancer, like high BMI, alcohol consumption, early menarche, pregnancy, this kind of thing, really the top dog still seems to be aging. It is by far the highest risk factor for breast cancer. And if you look at the distribution of ages at which people are getting breast cancer, you can appreciate in the United States that you get this kind of bimodal distribution. And that's interesting in and of itself, that you have sort of a younger and an older group. But when you layer on top of that some glimpses into other countries, like Japan, it's a slightly different age distribution. It tends to weigh in on the younger side. But when Japanese women were relocating to the United States in their teens and 20s, there's a change in the distribution, which for me sort of indicates that there's a strong interaction between age and environment and what makes a person susceptible to breast cancer. And the other thing that sort of struck me is that this older group should be largely preventable. That's modifiable risk if we understand biologically what's underlying that. And when we started the work over a decade ago, the most common refrain, why does an older person get more cancer, is because they accumulate more somatic mutations. And of course, that's an incontrovertible fact that we do accumulate more mutations in our cells at random as we age. But I think James DeGregory has made a good argument that what we're really looking at is susceptibility to cancer, not necessarily acceleration due to the accumulation of mutations. And there's a nice example in this paper that they published in eLife in 2019, where they basically normalize the incidence of cancer to the total population. And what you see is that, independent of some specifics in terms of incidence, age is a very dominant effect, even in cancers like CML, where you could argue are potentially due to one single mutation, the BCR-ABL translocation. So it's not necessarily the case that you have to accumulate multiple mutations in order to get more cancer, that there's something else happening. And James has they added in this paper, they added in a nice model that sort of is, their explanation is that you have the tumor microenvironment or the microenvironment of the tissue that is simultaneously changing during the aging process. And in young organisms, you have this very repressive environment that keeps bad cells in a less fit state. And then as you age, not only are you accumulating more mutations, but you're also, the sort of overall microenvironment is changing and becoming less repressive, and sort of changes the balance of fitness for a cell that may have accumulated a mutation. And so I do think it's useful to think about aging as increasing your susceptibility to cancer, and not necessarily sort of a process itself driving cancer. And so I do ask myself occasionally, why do we even age? And I'm going to tell you why I think we age. We're, all organic organisms living in the universe are subject to the same sort of physiochemical entropic drivers. So oxidation, glycation, ionizing radiation, demethylation, things that just sort of happen to organic molecules spontaneously because entropy is trying to drive us into dust. And so you have this sort of constant application of physiochemical drivers that, over time increases the effects of cellular aging. And then if you sort of accidentally damage some, like a protein or a gene that's involved in repair of some kind, you can accelerate and get degenerate outcomes. You can sort of accelerate the aging process. And this is our sort of this is our very simple model of a tissue with several cells. And these entropic drivers are basically at random causing damages. And you can't predict what kind of damage or where it's happening. So at the single cell level, we really can't predict how aging is going to affect you or affect a single cell. But you have things happen to some cells, like you cause something that drives them into senescence, and then maybe they start secreting these senescence-associated secretory factors, SASPs, which could then sort of have, it could take a single cell effect and exert a much wider outsized effect on the tissue. And then you could take these random single cell damages and integrate and convert that into a more stereotypical change. And we've been wanting to understand the basis of these, how it is that you have basically apparently random damage happening in the tissue, and yet at the level of the organism, even the population, the whole breast tissue or the whole tissue, and even or other whole organs, and then specific tissues within the breast or within a tissue or within an organ rather, are changing stereotypically. You have, grain and wrinkling of the organism. You have these sort of population-based measures that we expect to see. You have in the breast increasing of adipose tissue and decrease of connective tissue. And then what we have spent an enormous amount of time really focusing on over the last 10 years is what's happening in the epithelia. And really for us, the most pronounced change that we observed were changes in intermediate filament expression. That was sort of the first thing that we noticed in the luminal epithelia of older women. And as I'm going to show you, it actually goes much further beyond that. Nevertheless, this is, sort of, it has to be a case where you have essentially random damage that gets somehow, integrated into these very stereotypical outcomes. And we'd like to get to the bottom of that. And that's what we're trying to do. Our chief tool is we really try to work in human primary cell culture. And the reason that I started doing that from the outset was there's some, I think, important differences between rodents and humans that we just didn't want to kind of get into. And we decided to put our energy into trying to develop a fully human system, despite the deficiencies it will have. It's, you know, mainly in vitro, and you can kind of compare back to in vivo and stuff like that. So there's certain leaps in logic that are more difficult to make when you're not working in an in vivo system. But we're sort of placing our bet there, and we're working on developing that system. It was actually started by Martha Stamfer, and she started collecting breast tissue and trying to learn how to culture these normal cells in the 70s. And I think maybe the first tissue collected was the same year that I was born. And then I met Martha in around 2007 or so, and we started working together. And since then, we've added significant tissue to it. The original tissue that was collected from reduction mammoplasty, we know the age of the people, and we don't know much else. And then since coming to City of Hope, working with Vicky Seawalt and Lisa Yee, who are translational scientists who run the breast cancer high-risk clinic, we've been collecting additional normal tissue that's either average risk or high risk, depending on the germline sequencing. And it's a very robust system for, you know, a scientist that wants to study aging in a human epithelia, because it's a tissue that is not required for the individual's existence, but it is required for the species' existence, right? And so it's quite robust. If you do know how to grow the epithelial cells, you can pretty easily, starting from a vial of organoids. We can freeze back thousands of ampules at fourth passage, and a lot of these strains will go out 15, 16 passages. So it's really, you know, a very robust technology. We do a lot of work at fourth passage because kind of the gang is all there. All the cell types that we've tried to identify in vivo seem to be present in our fourth passage cultures. And you can do lots of cool things with them. You can make fairly simple bilayer cultures. You can make more complicated sort of clonally derived three-dimensional structures and reconstituted organoids. And we've done a lot of that, the latter with Zeb Gardner. And over the years, we've accumulated a fair amount of tissue and established a number of sort of primary strains. And I'm happy to share with any of you if you'd like them. We recently, this is just a little bit of advertisement, but we recently started looking at the ancestry because we want to see if we can start to address some of these ancestry-related questions. And these are strictly the City of Hope patients, and we find that it's actually quite a diverse collection of individuals. And I think as we grow this over time and do more ancestry analysis, it'll be a better tool for looking at questions of ancestry and how that affects biology. But that is, I think, early days. So when we started this work around 2007, 2008, there was not a ton known about, you know, how aging manifests in the breast tissue. Basically, it was known that there was increased fat, that there were discontinuities that appear in the basement membrane, which I guess one could expect would affect polarity. Other than that, you know, there were maybe 9-10 papers, and it was really discussing normal tissues were basically a control for cancer tissues. And so it wasn't specifically, people weren't specifically studying, like, during the aging process, what's happening in breast. And so using this, primarily this cell culture system, what we've learned over the years is that, you know, there's sort of a decrease in the proportion of tumor suppressive myoepithelial cells. There's a sort of an accumulation of dysfunctional progenitor cells. There's changes that seem to arise in molecules that are involved in bilayer organization. We did look at telomeres, and we don't find a lot of evidence that there's shortening as a function of age. This is sort of one of the boxes we wanted to tick off early on. But there is an interesting difference where us and Meeker, and I forget, there's one other group, that there is a difference between the length of telomeres in myoepithelial versus luminal cells. And in the same people, the luminal cells always have the shorter telomeres. But we don't again, see like a shortening as a function of age. And then I'm going to talk a lot more about this, but luminal cells start to look really basal with age. They take on properties, both protein and gene expression, that we would normally associate with the myoepithelial cells. And then more recently, we had a little pandemic paper looking at the immune milieu as a function of age and normal breast tissue. And we find, based on the sort of the concentrations of T cells, B cells, and macrophages, that there's more immunosenescence and a more immune suppressive milieu as women get older. And this is in the periductal region, so really almost adjacent to the ductal structures. So when we moved to City of Hope, that was actually quite important for me because we'd spent a long time just cataloging, what we thought were age-dependent changes in the mammary epithelium and in the breast tissue. I didn't really have any indication that those were really related to risk in some way. Was it just aging? And we could make some arguments about why we thought the changes were important to cancer risk, but we didn't have any clear indication that they were directly reported or related to susceptibility or cancer risk. So when I came here to City of Hope, Vicki Seawalt has been just an amazing collaborator of mine. And we've together collected a significant number of normal breast tissue from high-risk women, and they came through the clinic. So they have, sequenced monogenic germline mutations in BRCA1, BRCA2, PALB2, and a number of other genes. And we sort of focused on the big three, BRCA1, BRCA2, and PALB2, because those women have sort of a lifetime risk that's over 70%. So you would, I guess the linkage that we want to make is that we would expect these women to get breast cancer, whereas just in the average population, we can't necessarily have that expectation. So when we went and looked at the various age-related markers that we defined, and so on the right is sort of one of the chief ones that we look at, which is keratin 14 expression in the keratin 19 luminal cells. So in sort of average risk aging, we see that in young women, the tendency is for these markers to be kind of mutually exclusive. That you have beautiful keratin 19 positive luminal cells, and then you have keratin 14 positive myoepithelial cells. In older women, we see more of this, where we get sort of co-expression of keratin 14 in the keratin 19 positive cells and some changing in the architecture and things like that. What we find in the BRCA women is that much earlier, so like women in their 20s, even teens, who have elective prophylactic mastectomies, and we look at their tissue, we see this really quite a striking association or expression of keratin 14 inside the luminal cells. Another, we do a lot of cell culture, and for years, luminal cells don't grow well. Normal luminal cells don't like sort of our standard 2D cultures, and we sort of solved that early on, or started to solve it with the media that we design, and at some level, these, just culturing the primary cells is in and of itself a functional assay. And what we see is that the proportions of luminal cells in young cultures are always very low. As women get older, these are all average risk cultures, the luminal cells are more successful in these, sort of stiff culture environments. And you really see that in the high risk women, that the luminal cells are really quite successful, in some cases shockingly successful at growing in these environments. And so there are changes in fitness in these stiff basal environments as women get older. The luminal cells just become much more successful. And sort of on the left here is a summary of what we think are various biomarkers of aging in the breast, and then which of those also seem to come up much earlier in women who are at higher risk. And so this was a way of us linking, these age-related changes directly to susceptibility, more directly to susceptibility-related changes. Keratin 14, I'm going to talk about that more throughout the talk more later, but changes in intermediate filament expression generally are actually quite interesting, and we see a lot of that happening in the luminal cells. Another marker that I think it's quite remarkable, actually. It's ELF5, which is a transcription factor. It maintains ER-negative luminal cells. And, there's a lot of discussion in the aging field about biological clocks. And a lot of times it seems like they use liquid biopsy or whole tissue biopsy, and it's not clear to me what's happening. This was one of Masato Miyano's real brainchilds, where we were looking early on, and he noticed some tantalizing changes in ELF5 expression that he thought it was consistently lower in older luminal cells compared to younger luminal cells. And so we published this nice paper where just looking at the expression of ELF5 mRNA in luminal cells as a function of age actually gives you a pretty good clock. And then what really stands out for us is when we started to look at women who came through the high-risk clinic. So it didn't matter whether they were BRCA1, BRCA2, or probably 2 carriers. They were consistently sort of, that clock was accelerated about 10 to 40 years. And so this is another one of the sort of chief tools that we have. And we're interested in, I'm not going to talk about it, but we are interested in trying to apply this sort of in some clinical settings to see if we can use this and related clocks and risk assessment tools, and ideally like monitoring the effectiveness of breast prevention studies. But for now, it's just a useful marker. Less ELF5 typically is a phenotype of older luminal epithelial cells. So I've been talking a lot about luminal epithelial cells, and I'm going to keep doing that because I think these changes in the luminal epithelia are actually really important because it's, number one, the most pronounced, where we see the most pronounced changes in the epithelia. But there's a number of studies that point to the likelihood that luminal cells and maybe c-kit progenitor cells, which are also biased towards luminal cells, are breast cancer cells of origin. And so we really we're trying to get a deeper understanding of how these changes in the luminal cells are occurring. Rosalind Simon, who used to be in the lab and now she's at UCSF, is a brilliant bioinformatician. And she took the first stab in the lab of really trying to take a genome-wide approach to see just how profound these changes are. And the way we did it was we used FACS-enriched luminal or myoepithelial cells. And then we did a lot of sequencing and sort of both methylome and transcriptome analyses. And for us, the results were pretty shocking. In terms of differential gene expression, most of the change is in the luminal cells. 95% of it is we see sort of age differentially expressed genes happening in the luminal cells, not so much in the myoepithelial cells. And that was consistent with sort of our lower resolution work that we had done in the years prior, where we didn't see a lot of changes in myoepithelial cells despite our best efforts. DNA methylation changes. So we did this particular analysis with the Infinium 450K and EPIC arrays. So it's mainly genic regions. And we see that in almost, I think it's all but 4 differentially methylated regions are in luminal cells, where there's very, very few in myoepithelial cells. So there's like a shocking amount of change happening in luminal cells specifically, not so much the myoepithelial cells. We recently took this group of genes that's differentially expressed in the luminal cells and did a little gut check to see kind of what's going on in these very nice sort of single cell sequencing data sets from normal breast tissue. And it's satisfyingly consistent. So we looked at the data derived by Hall et al. In the Brugge lab, the Murrow et al. In the Gartner lab, and then Ni et al. in the Kessenbrock group. And basically, my label's moved, I'm sorry. But number one, like our sorting strategy basically identifies luminal versus myoepithelial cells. That sort of is consistent. We did all our mapping according to Kai Kessenbrock's mapping strategy. So luminal 1 cells are the ER negative cells, luminal 2 cells are the the hormone receptor positive group of luminal cells, and then the big red blotch is the myoepithelial / basal cells. And when we look for the genes that are going up in luminal cells with age, they show up in that signature shows up most prominently in the basal cells, so in the myoepithelial cells. The genes that go down in luminal cells with age also typically they show up more in the luminal cells. They're higher and they go down in the basal cells. So, and that's consistent between all the data sets. And for us, I think that's another pretty clear indicator that this notion where you have a loss of lineage specificity or loss of fidelity in the luminal cells that tends towards a more basal phenotype is really quite consistent and robust. And there's some other interesting things that we think we see in the data. We don't do, for those of you who know me, you know this, that we don't do single cell data. Single cell sequencing. And I think it could be interesting to talk about the differences that people are reporting, because almost any difference that we see, no matter how small it appears, using Wilcoxon statistics, tends to come out significant. And we're not really confident that that's a true reflection of what's going on. But this basal shift seems true enough that seems to be very consistent. So it really took quite some time to find changes that were affecting myoepithelial cells. And it wasn't like these, there wasn't a strong case that we could make that we were seeing these directional changes in myoepithelial transcription like we do in luminal cells. But Roz, figured it out. And what does seem to be happening independent of lineage is that there's an increase in variances. So if you take your favorite gene and you look at it in a cohort of young women, the tendency is that the window are very tightly controlled. So the expression is very tightly controlled. You look at the same gene in a group of older women, say women in their 50s and up versus women in their 30s below. In that older group of women, the interquartile ranges just kind of blow up. And so we see just generally increasing noise. And there have been some satisfying examples of this at the single cell level from Martin Correa and there's a group at Stanford, and it's just skipping my mind right now, in pancreas, but also in the mouse immune system. So this increases in variances seems to be an important aspect of aging. The genes or the pathways that seem to be affected both by differential gene expression in the luminal cells and also by differential variances are probably not, probably to no surprise, they are, things that we typically associate with cancer risk. We are trying to understand the basis of this increased variance, and our early studies in that lead us to think that there's an underlying epigenetic component. And so our reasons for thinking that are the following. So again, this analysis was done with the Infinium EPIC and 450K arrays. So we're looking at mainly genic regions. And what Rosalind did is took all of the methylated sites and ranked them from based on beta values from most hyper methylated to the most hypomethylated in the young women. And then we looked at the matching site in the older women. And what she finds is that the biggest changes are happening in the hyper and hypomethylated regions, And that's also where we see the biggest variances. So this larger plot here is looking at specifically the variances in DNA methylation level. And both in younger and older women, these sort of hemimethylated sites tend to have a lot of heterogeneity, and that makes sense. But in the older women, we really see the variances increase in places that would have been hypermethylated or hypomethylated in the younger group. And there's an enrichment of these changes at some interesting sites. So for instance, in the luminal epithelial cells, looking at where we see more methylation are in places, these green words or these green transcription factors are transcription factors you associate with luminal lineage. And we see more methylation at those binding sites in the older women, of the luminal cells of the older women. And you see kind of the converse happening at binding sites that we would normally associate with the myoepithelial lineage. So there's less methylation around the P63, TP73, and these TEAD binding sites. TEADs are the partner of YAPs or YAP1, TAZ. And so, and those tend to be enriched in young women and myoepithelial cells. So there's, you can see at this sort of global methylation level that there's like a switching of the epigenetic regulatory mechanisms. Dustin Shawns is a professor here at City of Hope, and he likes to geek out over transposable elements, which are really cool. And so, he's taught me a lot about them. And it's interesting that, that it seems like one could argue that the existence of a lot of the DNA methylation apparatus is to shut down transposable elements and keep them controlled. And so we decided to do a whole genome bisulfite sequencing analysis of young and old luminal cells to look to see if we could see any interesting changes in the intergenic regions, and sort of look where the transposable elements are hanging out. And what Dustin and Parijat Senapati found is that basically, if we look at the evolutionary age of these different classes of transposable elements, where we see methylation loss is skewed heavily towards these evolutionarily young transposable elements. And to get an example of why we think this could be introducing more variances into the system, over here is looking at sort of what the whole genome bisulfite sequencing tracks look like at a specific region, where we see reduced methylation, and you can see the smooth curves down here. And if we look at the corresponding ATAC seq, so looking at chromatin opening, we see that the chromatin opens where DNA methylation drops. And so this is an example of one transposable element site. And then here's a sort of another example of why we think there's a relationship between the changes that we see in our older luminal cells and then breast cancer in specific, where we see these green tracks are sites that are losing methylation in the older epithelial cells. And we see that in a number of breast cancers, ER-positive breast cancers, that they're also losing the methylation in the same sites. And then looking at sort of some normal tissue versus DCIS, we see some evidence that this might actually lead to active transcription. And there's a few genes here just to show you examples of correlations between transposable elements that are getting demethylated and expressed with nearby genes like BARD, PODI, ZNF92, and HPN. So we do think that there's an argument to be made that that these aging is causing a demethylation of certain transposable elements in luminal epithelial cells that leads to non-stochastic changes in gene expression that are maybe relevant to breast cancers. This is another looking at all of the sites where we see demethylation and open ATAC-seq signal. in our normal older luminal epithelial cells, and then mapping that onto this group of breast cancers. I think it's from Metabrick. And it's, it sort of satisfyingly clusters things into, you have a group here of basal breast cancers, but then you have a group over here of luminal cancers, and the majority of the opening chromatin sort of clusters the luminal type breast cancers together. So we think that these sort of epigenetic changes are important in understanding why we get sort of these increased variances as a function of age. And it seems like they are probably important to cancer initiation or susceptibility. So one, I think, important question then is like, what maintains luminal cell states? And how do we go from an apparently unchanged myo-ep, although now we know they're increasing variances, the direct neighbor is the luminal epithelial cell. A lot of the genes that are changing with age do seem to impact myo-ep luminal communication genes. So some time ago, we've been trying to figure out how to culture luminal cells for a long time, just so we could kind of start to get at this question of how these different genetic changes are affecting the maintenance of the luminal lineage. So in 2017, we published a paper where we showed that directly co-culturing luminal cells on the apical surfaces of myoepithelial cells is a great way to get them to get luminal cells to grow and also maintain sort of a more robust gene expression. And a more recent evolution of that, or I guess in line with that, Michael Todd-Hunter in the lab, he's kind of a crazy dude, and one time he just started to feed, instead of throwing away the really dense cultures, he just kept feeding them and feeding them and feeding them into these cultures that we call post-confluent cultures. And what really starts to happen is the myoepithelial cells dive underneath the luminal cells, and he keeps them in culture for a couple months. And what we see is that in these more complex tissue culture environments, in standard 2D, there's a lot of contact between the luminal cells and the plastic, and they don't like that. And correspondingly, we typically have lower levels of luminal cells and lower levels of c-KIT positive luminal progenitor cells. And it's really quite simple that just by letting them proliferate and sort of, I think, generate their own microenvironment, you really start to see profound increases in the proportions of luminal cells and the proportions of c-KIT progenitors. And it has to do with the importance of this sort of cell-cell contact microenvironment. So Masaru Miyano was the first in our lab to start playing with heterochronous cultures. And so sort of mixing together either young myoepithelial cells with young luminal cells, and then measuring the gene expression in the luminal cells, or old myoepithelial cells with young luminal cells. And so in the young version, we get robust, young primary-like luminal epithelial cell expression. When you switch to old, we see that the tendency is to shift closer to an old-like gene expression. And using our sort of Wunder marker, the ELF5, we see that in young, the luminal cells maintain high ELF5, but in young old, the luminal cells, the ELF5 is dropped down. You can see changes in ELF5 promoter methylation, but you have to wait a lot longer and it's more complicated. So I think there's an interesting relationship between the protracted exposure to a microenvironment and certain methylation states that we would like to understand better. But what we then went on to do is take all the genes in the myoepithelial cells where we see increased variances. There's a few I told you that were differentially expressed. We also had a mass spec set data set, and there were a few that we identified there that were differentially expressed. And basically went into old myoepithelial cells and modified them with shRNAs against all of these high variance genes or differentially expressed genes. And the idea was that if it's highly variant, that we would sort of re-regulate the N expression level. And I don't think it matters that it's up or down, but we have to prove that still. But just sort of bringing it into tighter control of these genes that were highly variant. Whereas other genes that sort of were just increasingly expressed with age, you would just drive that down. And what we found, to my surprise, is that the genes that were differentially expressed in the myoeps with age, which is just a few, we didn't really see any changes in the ability to maintain the young luminal phenotype. But when we brought under control the high variance genes, it seemed to be quite effective in bringing under control the aging phenotype in the young luminal cells. And so this is not something that I understand yet, but what I think is happening is in these high variance genes, we think they're highly connected genes. And so by sort of regulating one or two of those nodes, it may bring back into control some other important cell-cell communication networks, pathways. And this is something also that we're interested in further understanding, which is why we worked with Kai Kessenbrock to do a single cell study, which recently came out, and satisfyingly, they reproduced our original findings that there's sort of an increase signature in luminal-esque progenitor cells. And then there's sort of a loss of lineage specificity in the luminal cells. And we're hoping, through iterative work with Kai, that we can sort of get at some of the, what is the impact of these variances. But there are other effects, that Kai's data led us to study that are outside of the myoepithelial luminal conversation. And I'm just going to show you a little glimpse of that. So Kai's data found evidence of this, what he's calling a pre-cancer associated fibroblast signature that is increased in the higher risk breast tissue compared to the average risk breast tissue. And then Eric Carlson in the lab has been dutifully establishing primary fibroblast cultures from our, he basically isolates them away from the epithelial cultures just as a matter of cleaning the culture as we established the epithelial culture. But instead of throwing it away, he started to subculture it and look at it. And what he's finding is really fascinating. There's looking, sort of parsing the populations with this CD105, which is endoglin. He sees that there's a higher proportion of endoglin fibroblasts as women get older. And correspondingly, there's higher proportions of these endoglin expressing fibroblasts in the high risk tissue compared to the average risk tissue. And he's been doing some interesting co-culture experiments with these fibroblasts and c-KIT positive, c-KIT enriched luminal progenitor cells. And normally, like when we're looking here at the balance of keratin 14 and keratin 19 expression, it's sort of a young, normal c-KIT positive cell typically gives rise to more luminal-like cells, keratin 19 expressing cells, and fewer keratin 14 expressing cells. But when he co-cultures them, he skews that whole distribution more towards basal. And so what we think is going on is this is another sort of important step in understanding why we get this increasingly basal luminal population as women get older. So there's these direct cell sort of maintenance of specificity, and there might also be sort of a stromally imposed differentiation phenotype as well. These, expression in keratin is something that I think is actually really important. And I'm going to spend the last few minutes of the talk focusing on why I think those keratins are something that we need to pay more attention to in understanding breast cancer risk. So I'm going to start this section by talking about Lydia Son, who's a professor at Berkeley, and she's a physicist turned bioengineer, and her lab developed this beautiful tool called the mechano-node pore sensing device. And basically, compared to other ways of, that are sort of commonly used to measure mechanical properties of single cells, we think that this particular method both gives pretty high resolution at high throughput. So we can look at 500 cells per second and any sort of single cell suspension. And we published a paper together a while back where we showed that you can, based on the mechanical parameters that we calculate out of the device, you can separate luminal cells from myoepithelial cells, malignant from non-malignant cells, actually looking at different stages of the malignant progression. And we thought that this was the end of the story, looking at the mechanical parameters that separate young from old cells seem to be the time. So if you squish the cell, we measure the time it takes to recover its shape. And it seems like older cells do that much slower. Stefan Hentz is a postdoc in the lab, and he wanted to go a step further. And he said, there's a lot more interesting, there are more dimensions that we're ignoring. And so he built a machine learning classifier to look at whether we could, classify young versus old cells. And there is more than just recovery time. So this this blue line is basically looking at the area under the curve if we only consider recovery time. When we use the full feature set, he gets over 80% accuracy in this classification model. And what then turned out to be really interesting is we looked at, we looked at the high risk population, the high risk cells, so BRCA1, BRCA2 carriers, and invariably women with these monogenic risk mutations mechanically look older than their actual chronological age. So there's this sort of emergent mechanical property in that's associated with risk, we think, that might be useful and important to measure, but it also led us to some interesting sort of biological pathways. So because we had seen these profound changes in keratin 14 in the luminal cells, Stefan went in and just with our normal strains, either knocked down keratin 14 in older strains or overexpressed keratin 14 in young strains. And basically the expression level of keratin 14 determines the predicted age group. And it kind of works, when I initially was thinking about intermediate filaments and cytoskeleton, I thought, oh, if we add more cytoskeletal components, more keratin 14, that the cell would become stiffer, kind of like the way I think about actin. But actually the opposite was true, that the more keratin 14 we add, the cells become a little softer. So it's not sort of a stiffness related thing necessarily that that in this case, the intermediate filaments are affecting. And I suspect that it's the intermediate filaments are like a filamentous signaling platform, like it's a place for molecules to bind on. And so Stefan did a proteomics analysis of young and old luminal cells, young and old myoepithelial cells. That data is published and you can download it. And what we specifically wanted to look for were proteins that co-regulated with keratin 14. And I won't go into all the details, but it has a lot to do with Stefan's training, that he zeroed in on this molecule PEAK1, which is a tyrosine kinase. It's a non-receptor tyrosine kinase. He trained with Rolf Brecken, and Rolf, some of you may know, has a passion for DDR1. And at some point they showed that downstream of DDR1 is this PEAK1 protein. And so we knew that there would be a small molecule that we could take advantage of to sort of see what happens if we modulate this pathway. It does, this WGCNA plot that I showed you is shows pretty modest increase in peak-1. But we do see by RNA-seq, by mass spec, and looking at peak-1 phosphorylation with CyTOF, we think there's reason to believe that the protein increases with age, and it's sort of the pathway is activated. Looking at some immunofluorescence of the epithelia, I think it's really quite striking. And young women, it looks like there's very little peak-1, if any, in the ductal luminal cells. And then in contrast, in the myoeps, they actually have quite a bit, and it looks like it's on the basal surface. So there's probably some conversation happening with ECM that is affecting the polarity of that protein. And the older cohort of women, average risk women, we see that the luminal cells express a shocking level of the peak-1 protein. And then even in the young, very young, high-risk women, this is one that I looked at. This is a BRCA1 and 2 mutation carrier. But even just the regular BRCA1 carriers show this similar phenotype where there's just a lot of increased peak-1 expression in the luminal epithelial cells. And so then Stefan went and started making measurements. So if we add 7RH, which the thinking at the time is, we inhibit DDR1, and so we then inhibit peak-1 phosphorylation. And so you can see that that is in fact the case. And he shows that it made older cells look mechanically younger. Okay, so that's great. And then what we're slightly more excited about is when we looked at the sensitivity, sort of cell viability to 7RH. And generally what we see is that in older, cell cultures from older women, the luminal cells are selectively sensitive to blockade of the peak-1 pathway, and they actually die. So it's not that they kill the myoepithelial cells; it selectively kills the luminal cells, and in older women. And we're thinking about this as a way of imagining breast cancer prevention that started from this whole thing about what's changing with age, and now how can we take advantage of that. And we're excited and continuing to explore this possibility that you can selectively ablate old luminal cells, which would be the prospective, cells of origin for the age-related breast cancers. And so, just some thoughts in closing about how we can apply these lessons from what started out as trying to do aging biology in breast. So we could use biomarkers. We could use sort of aging biomarkers as risk assessment tools. And so one example of that is the ELF5 biological clock. We had a nice paper in Nature Aging looking at intermediate filament proteins. And so what is fascinating to me is that it doesn't seem to matter which monogenic risk mutation these women have. They have this accelerated aging phenotype. And so I'm wondering if there's, a good possibility that we could identify women who, they don't have a monogenic risk mutation that we understand, and yet there's, breast cancer in their family. Can we identify those women? And so that's something that we're now working towards asking. And then I think a general problem in the breast cancer prevention field is a lack of biomarkers that can tell us whether a given tested intervention is having a biological impact. Typically, people look at Ki67 expression and ER alpha expression, things like that. And we're excited to get the opportunity to test, and we're starting to talk with Carol Fabian about doing this, and of course Vicky Seawalt, and be able to ask the question: if someone is applying a prospective intervention, can we see in a reasonable amount of time changes in, for instance, ELF5 expression or changes in intermediate filament expression? And so that's a second avenue that we're interested in. And then, of course, the third is prospective targets for prevention therapeutics. And I showed you the example of PEAK1. I think it's, certainly based on Sandra's work, we should also be thinking about modulating the local immune milieu. And then just overall, this is what we've been trying to accomplish in the lab. And these darker blue arrows are areas where we've got active investigations underway, looking at sort of the communication between these different cell types and whether we can ultimately control the fate and phenotype of these luminal cells that we think are so important in the genesis of breast cancers. I just want to, my final statement is just going to be to echo what my former colleague and friend Arti Huria used to say in all of her talks. If you want to go fast, go alone. If you want to go far, go together. I have an amazing group of postdocs and students in my lab, and I have amazing collaborators who are willing to work with me. And so I thank all of them, and I thank all of you for your attention.
All right. Thank you so much, Dr. Labarge. The problem with doing this via Zoom is that you can't hear the thunderous applause you would be getting right now. And I might even be on my feet. That was phenomenal. Thank you. I mean, such important work. And we have a number of questions. So let's just jump right to that. Let's see. The first question came early during your talk. And I think you circled back to this at the end, but the question is whether the loss of specificity in keratin 19 refers to an intermediate cell state between luminal and basal.
Yeah, I think so. How do I answer this question? Yes, I think that is exactly what's happening. I think that when you're adding the keratin 14 into the luminal epithelial cell, this is, I mean, like really speculative. So please forgive me for that. But what I think is happening is you're really changing the signaling, the concentration of different signaling proteins. And so you are getting sort of an intermediate state. Really early on, we were tempted to look at whether there was something kind of like an EMT happening in the luminal cells. And I could never really find a strong evidence for that. It's like I see a hint of it here and there. Like sometimes I see an EMT gene changing, but not consistently. So yeah, I think that's kind of, we're getting the cells into some sort of a meta state of, luminal versus myoep.
Yeah, thanks. I mean, intuitively that makes sense what you say. I guess it remains to be seen.
Like plasticity. Like I think that one of the reasons there's so many arguments in the field over things like what's the mammary stem cell and stuff like that. It seems like it's the epithelia in the mammary gland, like plasticity is an intrinsic property of it. And yeah. Right.
Makes a lot of sense. I mean, your culture system can help pick up those dynamics, which I think is phenomenal, rather than getting a snapshot in time. So more questions. Let's see. Did you see if there's a difference for gene expression variances and DNA methylation between luminal progenitors and mature luminal cells in your data set for luminal cells in young versus old? And how do BRCA mutation or BRCA mutations affect gene expression in the luminal cells?
Good questions. So I'll start working from the back to the front. We haven't yet exhaustively analyzed our sort of gene expression variances in the high risk population. Some of that is in a paper that I referenced, a bioRxiv paper for 2022, and some of it is in there. But we need to do more work on that. As far as looking at sort of the c-kit cells in specific versus the more mature luminal cells, only recently were we possibly able to do that. And so that was Michael Todd Hunter's it was difficult for us using our standard culture techniques to get enough c-Kit cells to sequence convincingly, where we could do that kind of analysis. Now, Michael's technique of post-confluent cell culture actually makes that pretty trivial. And so that's something that we hope to look at down the road.
Nice. Zoltan Selassie is asking a thought question. Based on your results, how do you explain the shift in breast cancer risk for relocated Japanese population?
I don't know. I favor the barbecue hypothesis, and that is that culture and sort of food are intrinsically linked, and I think that our eating habits in the U.S. are stricken with overnutrition and things like that, and so that's I don't know. I really don't know. I think that interaction between aging and environment. That's a very clear, and I apologize. So.
Yeah, no. I think that is a question that's burning in the field. I think particularly Melissa Davis's work, where she actually followed chromosomes through the African diaspora and finding differences, in breast cancer risk, even among shared ancestry between women in Africa versus African American. I mean, it's the same type of story, but it says, there's a lot more that we need to understand about environment. Another question, and by the way, everyone is prefacing their questions with excellent talk and very interesting work, so you should know that too. There's a question about, would you link the increased expression stochasticity in patients with genetic predisposition when the predisposition genes have roles in DNA repair?
Sorry, could you?
That makes sense? Yeah. Would you link the increased expression stochasticity in patients with genetic predisposition?
Okay,
Maybe start there.
I'm gonna say what I think the question is asking. So, in these for instance, BRCA1, BRCA2 women, those are DNA repair genes, and so are we getting more variants because we're getting sort of more damage? And is that a reasonable way of restating that?
Actually, yeah. So it was rephrased in the Q&A. Is there evidence that increased expression stochasticity in patients with a genetic predisposition is related to loss of DNA repair, for example, in BRCA1/2 patients?
I don't know, but it seems to me like we sort of zoomed in on DNA methylation mainly because it's normally such a stable, sort of a stable mark, and I think there's a good reason to believe that these sort of, if you increase the amount of unrepaired damage, you are accelerating the process by, for instance, changing the regulation of epigenetic regulatory genes. Mm-hmm. And we do think we see some of that, and that's in another sort of study that it's in the queue of things that we need to write up. And we do see some changes in, for instance, CXXC5 and some TETs and DNA methyltransferase 3. And those changes seem to not only happen in the older women, but also in the high-risk women. So there does, and from our data, it seems like there's a link. I just,
Yeah. Interesting. Yeah. Well, probably time for two more questions. So another question, you kind of got to this, but it'd be interesting to hear whether cytokeratin 14 maintains E-cadherin localization as cytokeratin 19 does in some breast cancer cells. So an effect via cell adhesion disruption, et cetera.
Yeah, that's cool. I don't know. That's a great question.
Yeah. Cool. So I have another one of those thought questions for you. I mean, and it's something that comes up a lot in our work too, is that, we seem to have learned the most by stratifying the very young and the very old. And we get a lot of information from that stratification. And, if you look at work, for instance, that my friend and colleague, Rachel Friedman, has done looking at outcomes with age, there's sort of a bow tie sort of shape to the curve when you look at the hazard ratio for death, where the worst outcomes are seen in the younger and older patients, regardless of stage. So I guess my question is, what do we do about middle age? How do we, our age stratification is telling us some things about risk with age, but what do we do with the middle age group?
I'll tell you what I think we should do. I think we should look for markers that indicate accelerated aging.
Yeah.
Because typically, and it has to be more than just like one marker, and I think it needs to be fairly tissue specific. That's my guess. Also, some of you who know me know that I have for a very long time ignored the Middle Ages because there's so much variance there.
Yeah.
I feel like we need, a lot more of those samples to sort of power any kind of an analysis. And that's why we tend to look at things in sort of young versus old. Now I'm starting to think that, like increasing variances and things like that should show up a lot earlier in these middle-aged women who are actually going to be at higher risk. And that's where I would put my effort is to take as many sort of biomarkers of the aging process that are tissue specific and apply that in that group and look at people who are really the outliers.
Right. That seems to make a lot of sense. And like I kind of wave my hands. It's the same with the menopause question, right? Is how does menopause factor into all of this? And again, by the stratification, we can roughly say pre-post menopause. But yeah, I'm in your camp about, thinking of it that way. So unfortunately, we are out of time, but there are more questions in the Q&A. And if you don't mind, we can send those to you if you want to have some offline conversation with some of the folks who have joined. So
I'm happy to answer. Yeah.
Thank you very much. And once again, thank you for your time. Phenomenal talk, phenomenal work. Thank you for being here today.
Thank you very much. I appreciate the opportunity. Bye, everyone.
Take care. Bye-bye.